Hi, everyone. Welcome to Chapter 9, Molecular Biology. And you may hear those words, molecular biology, and think, oh no, she's bringing back the chemistry stuff that we were learning about in Chapter 2. And yes, that is true. We are going to visit some of the stuff that we learned about the biological molecules back in Chapter 2. But what I want you to know is that this chapter is the major reason why we have to learn about a lot of that chemistry stuff in the first place. Because in this chapter, we are going to come to understand exactly how it is that DNA brings about our traits. How do those letters in our DNA turn into characteristics? And along the way, we're going to learn about the cause of several genetic diseases and how our modern day medicine treats those diseases. So, what is molecular biology? Molecular biology is the field of biology that studies the structure and function of biological molecules, especially nucleic acids and proteins. So, you may remember from back in Chapter 2 that there are four major categories of macromolecules that living things are composed of, and nucleic acids and proteins are two of the four types of macromolecules. So, to warm up our brains and get us thinking back to Chapter 2, Part 2, I'd like to start off with a checkpoint where you tell me what the other two are. It's nucleic acids, proteins, what and what. And those other two macromolecules aren't going to be the focus of this chapter, but it is good to remember that they are there. So, why does molecular biology place a special focus on nucleic acids and proteins versus those other two types of macromolecules? What makes nucleic acids and proteins so important? Well, you may remember from Chapter 2 that nucleic acids are the informational blueprint for the cell, and one of the main nucleic acids is DNA. DNA tells the cells of the body how to make the physical structures that it needs and how to carry out its functions. And then proteins are what make up those structures and carry out those functions. So the nucleic acids are like the brains of the operation, and then the proteins are what take the action. So to summarize, nucleic acids direct the creation of proteins, and the proteins are what bring about the organism's traits. And I'll give you some specific examples to better understand this. So here's one example. Your hair is made up mainly of a protein called keratin. The instructions for making this keratin protein are written in your DNA. Some people have the alleles that encode a version of the keratin protein that coils up around itself, which creates curly hair. And some people have alleles that contain instructions for making a non-coiling version of keratin, which results in straight hair. And it all comes down to what the instructions in your DNA say about how to make this protein. Did the hair follicles make curly keratin, or they make straight keratin, or something in between? So the version of the protein that you have is the trait. Another example relates to a condition known as lactose intolerance. So people who are lactose intolerant can't eat dairy products because they're not able to digest and break down the main sugar found in dairy, which is lactose. People who are not lactose intolerant are able to digest lactose because their cells make a protein enzyme called lactase, and that protein is what breaks down lactose. But if your DNA doesn't contain instructions that tell your cells how to make this lactase protein, then your digestive system has no way to deal with lactose. And so if you eat dairy, you experience the indigestion and other symptoms that come with lactose intolerance. A third example relates to muscles and movement. So in muscle cells, there's a protein called actin, which plays a central role in allowing your muscles to contract. So every time you move, you can thank the actin in your muscle fibers for making it possible. There are different alleles encoding different versions of the actin protein, and studies have shown that elite sprinters and powerlifters tend to have a version of this protein that allows for faster, more powerful muscle contractions, which of course gives them an advantage in their sport. So hopefully these examples give you an idea of how our physical appearance and our bodily functions are related back to proteins and how the creation of these proteins is dependent upon what's written in our DNA. So this is why molecular biology is especially interested in nucleic acids and proteins. Nucleic acids give the direction, proteins take the action. But those three examples that we just talked about, they're related to some pretty benign traits. They may be interesting, but they're not serious, they're not life-threatening. And a major reason why molecular biology is so important is that it allows the medical field to understand and address serious diseases. So in this lecture, we're going to learn about molecular biology through the lens of several case studies of real people affected by real genetic diseases. And these are the people that will be the subject of our case studies. We're going to learn about their disease, the genetic cause of that disease, and in some cases, a treatment option that they used. But before we get to these case studies, we need to do a little bit more review from chapter 2, starting with DNA. And I know you've seen this several times recently, but we're just going to go through it again quickly. So DNA is a type of nucleic acid, which means that it is made up of strings of nucleotides. And every nucleotide is composed of three pieces: a sugar molecule, which is shown in green, linked to a phosphate group, which is shown in blue, and also a nitrogenous base, which is shown in pink. And this nitrogenous base varies. It can be 4 different things. Those 4 different things are thymine, cytosine, adenine, and guanine. And for another checkpoint, to review what we just learned in our previous chapter, I'd like you to tell me which of these nitrogenous bases are linked to each other when a double-stranded piece of DNA comes together. So fill in the blanks. In a double-stranded piece of DNA, thymine on one strand links to what on the other strand? And cytosine on one strand links to what on the other strand? So when you get those long strings of nucleotides together, what you're really looking at is a type of coded information. You've got A's, T's, G's, and C's all in a specific sequence. And in our last chapter, I told you that these letters are a language that spells out instructions for how the cell needs to go about creating all of its structures that it needs to survive. But how does that language work? So how does this series of letters actually translate into a physical protein, like keratin, for example, or lactase or actin? That is what this lecture is really about. But to get there, we need to revisit also what we previously saw about proteins. So in chapter 2, we learned that proteins are made up of monomers called amino acids. We also learned that amino acids have a chemical structure that starts with a central carbon atom that's attached to 4 separate things. There's a collection of atoms called an amino group, which is a nitrogen attached to 3 hydrogen atoms. There's a carboxylic acid group, which is a carbon attached to 2 oxygen atoms, a hydrogen atom at the top, and then something called an R group, which varies in its structure. And in humans, there are 20 different things that this R group can be. And here are a few examples. So for each of these R groups, the amino acid has its own name. When the R group looks like this, the amino acid is called serine. When it looks like this, it's called glycine. When it looks like this, it's called aspartic acid. And again, these are just 3 of the 20 possible protein building blocks. Here's the diagram where we can see all 20 of them. And there's a lot going on in this chart, so I know it can be intimidating, but like we did in chapter 2, I want to remind you that I don't expect you to know what each of these amino acids looks like. What I do need you to understand is that there's classifications for these 20 amino acids. They're grouped in specific ways because depending upon the structure of their R group, each amino acid is grouped into one of 4 categories. The nonpolar amino acids, which are shown in the yellow box, have an R group made up of atoms that are linked by nonpolar bonds. The polar amino acids, which are shown in the blue box, have an R group consisting of atoms linked by polar bonds. And then there are amino acids that have an electrical charge, meaning they've either lost or gained an electron from one of the atoms in the R group. So the ones in the green category lost an electron, so they have a positive charge, and then the ones in the purple category gained an electron, so they have a negative charge. Now, these terms polar and nonpolar, they might sound familiar, but in case you don't fully remember them, this is another concept we covered in Chapter 2, Part 1, which is different types of bonds. So ionic bonds are ones involving ions, where an electron is transferred from one atom to another. And that's what you see in the top row of this chart. The other two types of bonds are covalent, and in a covalent bond, the electrons are shared between the bonded atoms. But the difference is that in a nonpolar covalent bond, the electrons are shared equally between the atoms, while in a polar covalent bond, the electrons are shared unequally. And in the case of a polar covalent bond, the unequal sharing of electrons causes one end of the molecule to be somewhat more negatively charged and the other end to be somewhat more positively charged, which can lead to attractions between separate molecules. Okay, so getting back to our review of proteins, we also saw in Chapter 2 how proteins are formed when these amino acids come together and are linked to each other in a chain. So this is similar to nucleic acids and how nucleotides form long chains that become DNA strands. So we saw in Chapter 2 the short example of how just 3 of these amino acids can come together and can form a protein. And here we have the R groups highlighted in these boxes as we show the protein being formed. But in real life, those chains don't stay as long, stringy chains. Instead, they form into a complex three-dimensional structure. So here's a simplified illustration of the chain of amino acids in which each amino acid is one little bead. And in this animation, we can see an example of how the chain will cluster and condense and wrap around itself. This process is called folding. The final shape of the protein is what allows the protein to do its job. And the shape of every protein is different, and the way that it functions depends directly on the shape. So think of the keratin protein and how it can either bring about curly or straight hair, for example. Now in this animation, you see that the chain just starts folding up automatically. And that is actually a good representation of what happens in nature. The protein largely automatically folds itself into the 3D shape. But what causes that automatic folding? Well, there are a variety of forces that cause the protein to take shape, but they all relate back to the different amino acids that are found in the chain. Because as we know, each amino acid belongs to a different category depending upon the chemistry of its R group. Positively charged amino acids are going to be attracted to negatively charged amino acids. So if you have a positively charged amino acid at one spot in the chain and a negatively charged amino acid at another spot, they might try to find their way to each other based on that attraction, and that causes folding. Also, polar amino acids are attracted to other polar amino acids because remember, they have this uneven electron distribution, which leads them to have somewhat positive and somewhat negative ends. And in Chapter 2, we saw another example of how this works in water molecules. Water molecules are polar, and the negative end of one water molecule is attracted to the positive end of another water molecule, and that holds them together. So this is how the attractions work between polar amino acids as well, and this also contributes to the folding of the protein. And while we're on the topic of water, another force that drives protein folding is the fact that polar amino acids and electrically charged amino acids are hydrophilic, meaning they love water. And nonpolar amino acids, on the other hand, are hydrophobic, meaning they hate water. They're water-fearing. And this gets back to 2 of the properties of water that you learned about in Chapter 2, which is that water easily dissolves ionic molecules and other polar covalent molecules. So you may remember how we showed that the attractions that exist between water molecules can be substituted for attractions to other molecules that have full or partial charges. So, for example, if we drop a molecule of salt into water, it dissolves easily because it fits right into this network of attractions. But in the case of nonpolar molecules, which don't have those positive and negative ends, they form no attraction to other molecules, and so they'll just separate themselves from water. And this is what we see happen with oil and water. Oil is nonpolar, it's hydrophobic, it separates from water. So this is how the nonpolar amino acids behave as well. They want nothing to do with water. And yet our bodies are made of water and our cells are filled with water. So when a protein folds, it will not only be driven by electrical and polar attractions between amino acids in the chain, but also the desire of the hydrophobic non-polar amino acids to hide themselves away in the center of the protein where they won't have to be exposed to water. And then the polar amino acids can sit on the surface of the 3D shape and enjoy those interactions with water. Okay, so that wraps up our review stage. We've reviewed nucleic acids, we've reviewed proteins, we understand what proteins are made of and how they fold into 3D shapes. So now we can get down to business. So let's start in on our studies of genetic diseases. We're going to begin with Victoria Gray. Victoria is our case study for a disease called sickle cell anemia. Sickle cell anemia is a genetic disease caused by an abnormality in the protein hemoglobin. Hemoglobin is a protein that is produced in our red blood cells. Its job is to pick up oxygen in the blood vessels that pass through your lungs and then carry that oxygen through the bloodstream to various parts of your body. So you may remember how the cells use that oxygen in chapter 4 when we learned about cellular respiration and how your cells create energy for themselves. So that's what hemoglobin normally does. It is the carrier of oxygen. But the abnormal version of the hemoglobin protein that is produced in people with sickle cell anemia causes their red blood cells to become sickle-shaped. A normal, healthy red blood cell, which you see on the left side of this picture, is nice and round and plump, but sickled red blood cells, which you see on the right, have this crescent shape, which makes it more difficult for that blood to circulate. The sickled cells can become trapped in blood vessels, and this leads to a variety of symptoms, including anemia, attacks of pain, swelling in the hands and feet, organ damage from restricted blood flow, and stroke. The life expectancy for people with sickle cell anemia is 40 to 60 years. And in the United States, this disease affects about 1 in every 3,300 people. And it's much more common in people of African descent. About 80% of people affected by this disease are currently in sub-Saharan Africa. So let's talk a little bit more about this hemoglobin protein. The gene for creating the hemoglobin protein is located at 11p15.4, which, to remind you, means human chromosome 11, the P arm, which is the small arm, and site 15.4 on the P arm. So here I'm showing you both copies of chromosome 11, because remember, each chromosome comes in a set of 2. One comes from your mom and one comes from your dad, and each one of them has a hemoglobin gene on it. The gene for hemoglobin is 1,608 nucleotides long, and those 1,608 nucleotides spell out the recipe for making the hemoglobin protein. So this blue and red thing you see here is a model of what the protein looks like when the chain of amino acids is made and then when it properly folds. But how do your cells do this? How do they read the information in the gene and turn it into the thing that you see on the right-hand side of the screen? That's what we're going to walk through now together. So as you all know, our DNA is found inside of a dedicated compartment in our cells called the nucleus, and the DNA has to remain there. Each cell only has one master copy of the DNA, and that DNA has to stay in the nucleus where it can remain safe and protected. And actually, the DNA doesn't look like this. It's not in chromosome form, because as you'll remember from our chapters on mitosis and meiosis, the DNA doesn't wind up into chromosomes until the cell is about ready to divide. So in reality, the DNA looks like this instead. It's in the form of chromatin. So the DNA is in the nucleus, and that's where the information is located. But you also may remember from earlier in the semester that proteins are not made in the nucleus. They're made by ribosomes, which are mostly found in the cytoplasm. And I say mostly because not all ribosomes are drifting through the cytoplasm. Some are actually attached to one of the cell's organelles. So for our next checkpoint, I'd like you to think back to chapter 3 and tell me which organelle that is. Which is the one that has ribosomes embedded in its membrane? So regardless of whether we're talking about a ribosome in the cytoplasm or one that's embedded in this organelle, the information that is carried in the DNA needs to somehow be transported out of the nucleus and to a ribosome without moving the DNA itself. And the way that the cell accomplishes this is through the process of transcription. Transcription is the name of the process by which a mobile copy of the information within a gene is made. While the original gene is made of DNA, the copy is made of a different molecule called RNA. So, like DNA, RNA is a type of nucleic acid. The name DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And as far as biological molecules go, they are very similar to each other. So here we can see a comparison between a DNA building block on the left and an RNA building block on the right. We can see that they're both made of nucleotides, and those nucleotides each have the same basic structure of containing a phosphate group, a sugar, and a nitrogenous base. And the phosphate groups are totally identical to each other. But the other parts of the nucleotide are a little bit different. The sugar component of a DNA nucleotide is called deoxyribose, while the sugar component of an RNA nucleotide is called ribose. And on the level of their molecular structure, this really translates to there being just one less oxygen atom in deoxyribose compared to ribose, which is why the one on the left found in DNA is called deoxyribose. It has one less oxygen. And then the nitrogenous bases are a little bit different too. So as you know, DNA nucleotides contain nitrogenous bases adenine, guanine, cytosine, and thymine. But in RNA, the possible nitrogenous bases are adenine, guanine, cytosine, and uracil. So instead of thymine being an option in RNA, there's uracil instead. And then the last difference is that, again, as you know, DNA molecules pair up with each other and they're double-stranded, but RNA molecules tend to be single-stranded. They stay solo. Okay, so getting back to this business of making proteins, transcription will take the DNA gene and make a copy of the information in the form of RNA. This copy is created by a special enzyme called RNA polymerase. So let's see a little animation of this being done. Here we have our DNA shown in blue, and this is a very simplified drawing of DNA. We can't really see all the parts of the nucleotides. We can't see the sugars or the phosphate groups. Really, the important part to acknowledge in this drawing is the nitrogenous bases, which are the letters that link the two strands. So that's the level of simplicity that you're seeing here. We have two strands that are wrapped around each other, and in the middle, you can see that there's a section here where the strands have been separated from each other, and you can see the letters representing the nitrogenous bases on each strand. And separating the DNA strands is the first thing that RNA polymerase does when it arrives on the scene. So RNA polymerase is shown here in purple, and this enzyme has to separate the DNA strands because it needs to be able to read the letters. So it will identify the gene that needs to be transcribed, and then it will separate the strands, and then it will choose just one of the strands to serve as the template from which the copy is made. And it'll start building an RNA molecule that matches the DNA molecule. And when we say matches, we don't mean that it adds the exact same letters, but rather it adds the complementary partner of each letter. So you know by now that in the DNA molecule, A pairs with T, G pairs with C. So when RNA polymerase sees the letter C, it will add a G to the RNA strand that it's creating. So C's turn into G's, G's turn into C's, A's turn into not T's, but rather U's. Remember, there's no thymines in RNA, there's only uracils. So where A would have paired with a T in DNA, when it's pairing with RNA, it pairs with a U. And then every time the enzyme sees a T, it will build an A, and so on and so forth, continuing to add letters until it reaches the end of the gene. Okay, so now let's test your ability to predict what RNA polymerase does with the hemoglobin gene. The first 21 nucleotides in the gene for hemoglobin are what you see here on the screen. So for this checkpoint, I'd like you to tell me when RNA polymerase copies this initial portion of the gene, assuming that this is the sequence of the strand that serves as the template, what will be the sequence of letters in the RNA molecule? Okay, so now we understand how transcription works and how the DNA gets copied into RNA. And this piece of RNA that comes out of the process of transcription is called messenger RNA. It's called messenger RNA, or abbreviated mRNA, because it carries the message found within the gene. This is the thing that can leave the nucleus. This RNA, messenger RNA, can leave the nucleus and travel to the ribosome. And just one thing to keep in mind, the little cartoon here is very inaccurate because genes are hundreds or thousands of nucleotides long. So this is just a tiny snippet that we can follow to give us an idea of how the process works. So we have our mRNA. It's still in the nucleus along with the DNA that it was copied from. We know that it needs to get to the ribosome, but before the mRNA can do that, it needs to be processed before exiting the nucleus. What processing means is that certain edits are going to be made to the messenger RNA before it leaves the nucleus. And when we say edits, we mean that some sections of the mRNA are going to be physically cut out and removed, and the remaining sections will be stitched together. This is accomplished by proteins in the nucleus called SNRPs. These SNRPs have a very long name that ends up being abbreviated to SNRNP, but biologists just agreed to pronounce them SNRPs, which is very cute, and I'm a big fan of it. So these SNRPs will actually look through the messenger RNA and they'll read the letters, and they recognize specific sequences of letters. And when they see those sequences, it will prompt them to attach to the messenger RNA and actually cut through the strands at those points. And then the SNRPs will remove the sections that they cut out. The parts that get cut out are called introns, and the parts that remain are called exons. And the exons get stitched back together into one seamless, cohesive strand. So it's the exons that move forward to the next step. Only the exons contain useful information about how to create the final protein product. Okay, so now we've created our mRNA, we have processed our mRNA, and now the mRNA is going to leave the nucleus and travel to one of the ribosomes in the cytoplasm where the protein will be made. So let's take a moment and put this back in perspective of the disease case study that we're talking about. We're talking about sickle cell anemia, where the affected protein is hemoglobin. On the next slide, I'm going to show you the sequence of the final processed messenger RNA that leaves the nucleus to be turned into hemoglobin. Okay, so this is the sequence of letters. Admittedly, it's not much to look at this point because we still don't know how this turns into a protein. But that's exactly what's going to happen in the final step, which is called translation. So during translation, the ribosome uses the information in the messenger RNA to create a protein. It will read the mRNA sequence in sets of 3 letters called codons. So if this is our mRNA sequence, these first 3 letters are a codon, the next 3 are a codon, the next 3 are a codon, and so on. Codons are like the words of the nucleic acid language. But we need to translate that language into a different language, which is the language of proteins, which are made up of amino acids. So what's going to happen is each codon gets turned into one amino acid. Now, to remind you, there are 20 different amino acids. Here they are. And biologists know exactly which codons translate into each amino acid. That information is all summarized and laid out in this table right here. This is called the codon table. It shows you every possible codon, meaning every possible set of letters consisting of A, U, G, or C, and what they translate into. The way that you read this table to figure out the translation of a specific codon is by starting on the left-hand side. You ask, what is the first letter of your codon? So you find the row for that letter, and then you turn your attention to the top of the table and ask, what is the second letter of your codon? And you find the column and the box where it intersects with the row corresponding to the first letter, and that's where you'll find your codon and find its amino acid. So the names of the amino acids are shown in the main part of the box, and the list of codons that correspond to them are shown in those smaller white boxes. And we're going to practice using this table in just a second. Before we do, though, let's see how the ribosome works its magic and turns these codons into protein. So here we have our messenger RNA strand, fresh out of the nucleus, and then this big brown blob that you see is the ribosome. And you can see that the messenger RNA is sort of threading through the ribosome. And the first codon in the sequence is AUG. You can see it highlighted in the red box. The ribosome will read AUG, and upon reading AUG, it will bring in the corresponding amino acid. The amino acid will be delivered by a molecule called a tRNA. A tRNA stands for transfer RNA. Transfer RNA molecules are short strands of RNA that carry and deliver amino acids to the ribosome. They have a little sequence of 3 RNA letters, which you can see in the darker green color at the top of the tRNA. And these match up with the 3 letters found in each codon. So in our messenger RNA, we have AUG, and that pairs with UAC in the tRNA. And this tRNA is carrying the amino acid methionine. That's what the little purple thing at the bottom is supposed to represent. The methionine is the amino acid. MET stands for methionine. And if we cross-reference what's going on here with our codon table, we will see that AUG is listed here as translating to methionine. We'll also see that AUG gets a special little designation as the quote-unquote start codon. AUG is almost always the codon that is located at the very front of the mRNA sequence. So the first amino acid to get incorporated is methionine. And this is what methionine looks like as a molecule, shown in the red box. In reality, methionine is not a little purple blob. That's just how we're showing it in our animation. So getting back to that animation, the ribosome has called up a tRNA molecule that is the right match for AUG, and it's carrying the right amino acid. The tRNA will enter the ribosome and match up with that first codon. Then the ribosome looks at the second codon. In this case, it's GGA. It will call up a tRNA molecule that's a match for GGA, which has CCU in it, and it will be carrying the amino acid glycine, which is what the GLY stands for. This tRNA will then enter the ribosome alongside the first one, and then a new bond will form, which will link the 2 amino acids to each other. Now, when we get to the next codon, The ribosome wants to read that codon, but it's running out of room to bring in more tRNA molecules. So it has to kick out one of the existing ones. So it'll scoot down the line and it will let that first tRNA go. But that tRNA will leave behind its amino acid, the methionine, which is now attached to the glycine. And now there's enough space to bring in the next tRNA, which is carrying an amino acid called cysteine. And a new bond will form to link that cysteine to the glycine in the first spot. And the process continues to repeat in this manner. The ribosome moves down, it kicks out a tRNA to make room for a new one. The new one comes in, it delivers its amino acid and forms a new bond, and so on, until eventually the ribosome will reach the end of the gene, and it will know this when it reaches a stop codon. There are 3 sets of letters that are referred to as stop codons. You can see them highlighted here in red boxes: UAA, UAG, and UGA. And when the ribosome reaches one of these, it will stop translating, and instead of bringing in a tRNA, it will just release the string of amino acids that it has built. So our final product is going to be a chain of amino acids, which I've shown here on the left, highly simplified. And then, driven by a combination of all those forces we talked about earlier, the polar attractions between amino acids, the electrical attractions, the hydrophobic effect where nonpolar amino acids don't want to be around water, that chain will spontaneously fold itself into the shape of the protein, which in this case, remember, in this particular case study, we're talking about the protein hemoglobin. But this is how it works for all proteins in general, not just hemoglobin. But we do want to refocus our attention on hemoglobin again in this checkpoint. Using the RNA sequence that you came up with in checkpoint 4, along with the codon table shown here, what would be the first 7 amino acids that the ribosome adds to the chain when creating the hemoglobin protein? So we now have an understanding of the processes that cells go through to take the information in their genes and turn it into proteins. And armed with this knowledge, we can now learn about and truly understand the cause of sickle cell anemia. How is the version of hemoglobin made by people with sickle cell different from the normal version? Individuals with sickle cell anemia have a mutation in the gene for hemoglobin on both of their chromosomes, not just one, but both. A mutation is a change in an organism's DNA. So on the next slide, I'm going to show you the sequence of letters of the DNA template strand for the normal hemoglobin gene. So this is the same thing that we saw previously. It's just the original DNA instead of the messenger RNA. And now I'm going to switch it to the gene sequence for sickle hemoglobin. Did you see the change? I'm going to switch it back and forth again one more time. Here's the original. Here's sickle cell. If you didn't catch it, I'm going to point it out to you. This A right here that I've highlighted is the only thing that's different. In normal hemoglobin, there is a T here. In sickle hemoglobin, there's an A. And I'm going to pull this first sequence of letters out and show you so that you can see it more clearly. So on the top is the sequence of letters at the beginning of a healthy hemoglobin gene. And on the bottom is the sequence of letters at the beginning of a sickle cell gene. And the mutation is bolded. You can see the T on the top and the A on the bottom. And I don't know about you, but even after all my years in biology education, it still strikes me as so crazy that changing just one letter can have such a big effect. But it's true. All it takes is one mutation like this to cause a genetic disease. Mutations like the one seen in sickle cell are called point mutations. A point mutation is when a single nucleotide is changed, deleted, or added. It's a change that happens at just one point in the DNA. These are different from chromosomal mutations, which we learned about previously. Chromosomal mutations are changes that happen to DNA on a chromosome scale, so an entire chromosome being added or deleted. This includes the aneuploidies that cause, for instance, Down syndrome or Klinefelter syndrome. So how is it that changing one letter can cause a whole disease? Well, let's see what happens when this protein gets made. First, as we know, the DNA gets transcribed into RNA, which gives you these sequences. And then in the normal allele, the messenger RNA would contain an A, but in the sickle cell allele, the messenger RNA contains a U. And then I'm going to ask you to complete the next step, which is the translation of the messenger RNA. How does the mutation change things in the protein? So in this checkpoint, which amino acid does the 7th codon translate to in the healthy allele? You actually already figured this first part out in checkpoint 5. Look back at your answer for checkpoint 5 and see what you put for the 7th and final amino acid. The 7th codon is the one affected by the mutation. And then tell me what this 7th codon translates to in the sickle cell allele. Sickle cell allele is GUG instead of GAG. How does that change things? And I'm going to put the codon table up on the next slide so that you can pause it there and solve that. So hopefully by now you figured out that changing the letter in the 7th codon changes the amino acid. Point mutations that result in one amino acid being substituted for another are called missense mutations. And I'm going to give away the answer here, but it's important that we talk about the nature of this missense mutation because it's really at the core of the disease itself. Hopefully what you found is that the original healthy gene gives you the 7th codon being glutamic acid, and the sickle cell allele has the 7th codon as valine. I've highlighted both of those in the amino acid chart on the right-hand side. So what's the big deal? There's hundreds of amino acids in hemoglobin. Why does it matter that we change this one? Well, it has to do with the category that each of these amino acids belongs to. The original amino acid, glutamic acid, as you can see, belongs to the negatively charged category. But the sickle cell hemoglobin allele, the new amino acid, the mutated one, valine, is nonpolar. And if we think about what we learned about the forces that influence protein folding, we know that electrically charged amino acids, like glutamic acid, play well with water, but nonpolar ones do not. They want to avoid water. So we went from something that likes water to something that hates water. So whereas in normal hemoglobin, the glutamic acid, which is shown here in green, and it's actually labeled in this diagram as glutamate. Glutamate is another name for glutamic acid. So whereas in normal hemoglobin, this glutamate is perfectly happy to sit on an exterior fold of the protein and interact with water, when it changes to a valine, suddenly valine is very much not okay with being exposed to water. In fact, valine wants to avoid water so much that two separate hemoglobin proteins will clump together, and their valines will get together to try to shield themselves on the interior of the clump and separate themselves from water. And it's not just the case that only two hemoglobin proteins will do this. They'll get together in very extensive clumps and form these filaments of proteins, which is not how hemoglobin is supposed to behave. You can see normal hemoglobin proteins on the left and then the clumped version on the right. And these filaments of proteins inside of the red blood cells, this is what causes them to assume that sickle shape, which brings about the blood vessel blockages and all of the associated symptoms of the disease. All of this because of one letter, one spot in the DNA where there's an A instead of a T. So, how do you treat this? Genetic conditions aren't like infections. When you catch an infection, oftentimes it can be treated with medications and you can be cured of it. But genetic diseases are written into your DNA. How can you cure a disease that's written into the DNA? Until very recently, curing diseases like sickle cell was simply not possible. It was simply a matter of living with the disease and managing the symptoms as best as you could. But what if doctors could rewrite your DNA and correct that mutation? This is what happened for Victoria Gray. Gray was the first patient ever to receive gene editing treatment for her sickle cell. To understand how this works, you first need to know that our red blood cells are created from the cells of our bone marrow. So, some of Gray's bone marrow cells were removed and the sickle cell hemoglobin mutation was corrected using a gene editing technique called CRISPR. Then, the edited cells were infused back into her body and allowed to multiply into healthy red blood cells. And since her treatment, which took place in 2019, Gray has been free of symptoms. She was part of an experimental cohort as the FDA was going through the process of testing this procedure. And then later in December 2023, the FDA finally approved the treatment, which means that it's now available to others. So this is truly a remarkable development in medicine, to be able to truly treat the underlying cause of genetic diseases. However, that doesn't mean that everything is rosy about this therapy. There are major concerns about the equitable availability of the treatment, because as of the recording of this lecture, it costs $2.2 million to undergo this procedure, because it's very complex. It's basically like getting a bone marrow transplant with a bunch of other sophisticated technology built in. So while this treatment is without a doubt a major breakthrough, there are still big drawbacks in its lack of accessibility, especially in light of the fact that most sickle cell cases are in sub-Saharan Africa and developing countries where its affordability is basically out of the question for most regular people. We'll end our discussion of sickle cell with a checkpoint. We learned that individuals affected by sickle cell anemia have a copy of the sickle cell allele on both of their chromosomes. Heterozygous individuals who have one copy of the sickle cell allele, meaning they have the healthy version on one chromosome and the sickle cell version on another, these people have a condition that is referred to as sickle cell trait. And while they don't have full-blown sickle cell anemia, elite athletes with sickle cell trait are more likely to experience heat stroke, severe muscle breakdown, altitude sickness, and even sudden death when exercising. So they don't have full-blown sickle cell, but they're also not all the way normal. Which pattern of inheritance that we learned about at the end of chapter 8 describes a situation where a heterozygous individual has a phenotype that is intermediate between the dominant and recessive traits? So that wraps up our discussion of sickle cell. But sickle cell is just one disease, and missense mutations are just one type of mutation. We know that missense mutations result in one amino acid being substituted for another. But it's also possible for point mutations to result in other outcomes. For example, silent mutations cause no change in the resulting amino acid. And this is possible because there are multiple codons that give you the same amino acid. For instance, if the codon CCU changes to CCC, the original amino acid is proline, but the new amino acid is still proline. And you can confirm this by looking at the codon table. So there's a change in the DNA, but not in the resulting amino acid. That's why it's called a silent mutation. The mutation doesn't actually show up in the protein. There are also nonsense mutations. Nonsense mutations are ones that cause an early stop codon to be created. So, for example, if you change the codon UAC to UAA, the original UAC would normally give you the amino acid tyrosine, but the new codon, UAA, that's a stop codon. And so instead of creating the rest of the protein, the ribosome will stop at that point and just ignore whatever comes afterward. So for obvious reasons, nonsense mutations can be pretty devastating. They result in an entire section of the protein not being made, which of course affects the way that the protein folds and its structure and how it performs its function. And it's a nonsense mutation that is relevant to our next case study, which is the one of Ryan Benton. Ryan Benton lived with a disease called Duchenne muscular dystrophy, which is often abbreviated DMD. So, what is DMD? DMD is a genetic disease caused by an abnormality in a protein called dystrophin. Dystrophin is produced in your muscle cells, and it's part of a set of proteins that connects the cytoskeleton of your muscle cells to other body tissues. So in this illustration, the dystrophin is shown as the fiber in red, and the dystrophin allows the contraction of your muscles to translate into movement by linking the muscles to other body parts. And this goes not just for your voluntary movements that you do consciously, but also for your involuntary movements that things that are involved in your vital functions that you might take for granted, like breathing or the beating of your heart. Symptoms of DMD include progressively worsening muscle weakness, mobility loss, and paralysis. Eventually, the muscles that are involved in controlling vital functions like breathing and swallowing become affected, and the most common way that people die from DMD is respiratory failure. The life expectancy is 28 to 30 years. So what mutation causes DMD? Well, the gene for dystrophin is located at XP21. This means it's found on the X chromosome, one of the sex chromosomes, the P arm, and site 21. And the dystrophin gene is the largest human gene yet documented. It is 2.2 million nucleotides long, with 79 total exons that get stitched together during the processing stage. And together, they contain instructions for making this protein. Unlike sickle cell anemia, there is not one single mutation that causes DMD. There are a range of different mutations. But a common cause is a nonsense mutation, which, remember, we just learned that a nonsense mutation is one that creates a stop codon too early, where there shouldn't be a stop codon, which leaves a large portion of the dystrophin gene untranslated. So normally, the stop codon comes at the end of those 79 exons. And in this illustration, it shows a representation of the normal dystrophin gene, with the stop codon being the little black asterisk on the right-hand side. But some people have a nonsense mutation relatively early in the gene. And so the location of the nonsense mutation is shown in the lower portion as a little red asterisk. And in people with this nonsense mutation, everything after that point is not read by the ribosome. So the majority of the instructions for making the dystrophin protein are simply ignored, and all of those critical amino acids are left out. So naturally, this creates a non-functioning protein. But that's not the only cause of DMD, because while some people with DMD have a nonsense mutation, others have what's called a frameshift mutation. What is a frameshift mutation? A frameshift mutation is caused by the insertion or a deletion of nucleotides in the DNA sequence of a gene. So we're not just swapping a letter out, we're actually adding in or subtracting letters. And this changes the way that the ribosome reads every codon downstream of that mutation. The way the ribosome reads the mRNA in sets of 3 is called its reading frame. So here I have framed each codon in a red box. And then down below, you can see which amino acid each codon is translated into. But let's say one of the letters gets removed from the mRNA molecule. So let's say, for example, this adenine right here is missing. And I don't mean that there's a gap, I mean there's nothing between the U and A anymore. Instead, they're right next to each other. In that case, it would change the way that the ribosome reads the codons. It shifts the reading frame, which is why it's called a frame shift mutation. So again, here I framed the sets of 3 letters, and below it, you can see the amino acids that each codon translates into. And if we then do a side-by-side comparison of the original mRNA, which you can see on the top, and the mutated mRNA, which you can see on the bottom, the first 3 codons are the same. We've got methionine, glycine, cysteine. But then comes the point where the deletion took place. And everything after that point of deletion is different. Different codons, different amino acids. And so unlike a nonsense mutation, the ribosome continues translating, but it's just creating the wrong stuff. It's creating a bunch of junk that's not supposed to be part of the protein at all. And the ribosome doesn't know any better. It doesn't know that there's a letter missing. It's just doing its job and taking the reading frame at its face value. So those are the 2 ways that DMD can come about. Either a nonsense mutation or a frame shift mutation in the gene for the dystrophin protein. Now it's time for another checkpoint. So for this one, I'll have you think back to chapter 8 again and fill in the blanks. First, because the gene for DMD is carried on the X chromosome, it is considered a what trait? And second, are males or females more susceptible to genetic diseases carried on the X chromosome? So just like we did with our first case study, we'll wrap this one up by talking about a little bit about Brian Benton, his treatment option, which he was the first DMD patient in the US to receive what's called a compassionate use exemption from the FDA to be treated with stem cells for his condition. A compassionate use exemption is where the FDA allows someone to gain access to a treatment that has not yet been studied or approved by the FDA. So the FDA can't confirm whether the treatment is safe and effective, but they allow it in cases where an illness is terminal and people have no other options. So Ryan received infusions of stem cells that were derived from umbilical cord tissue. These stem cells lack the DMD mutation, and stem cells are able to divide into many different types of tissues, including muscle tissue. And tests showed that the stem cell therapy resulted in higher levels of healthy dystrophin and an improved quality of life for Ryan. Unfortunately, this therapy is not a cure. It provides a temporary infusion of healthy cells, but it doesn't correct the underlying mutation. And so in 2015, Ryan passed away at the age of 37 after having received 6 years of stem cell therapy. The therapy likely contributed to him living much longer than the average life expectancy for someone with DMD, which is only 28 to 30 years on average. This brings us to our third and final case study, which is Sam Burns. Sam Burns suffered from Hutchinson-Gilford progeria syndrome. And this photo was taken when Sam was age 17, not long before he passed away from complications of old age. And no, that's not a mistake. He died of old age. Hutchinson-Gilford progeria syndrome is a disease that brings about rapid aging, and it's caused by an abnormality in a protein called lamin A. Lamin A is part of a larger network of proteins that make up a structure called the nuclear lamina, which sits on the inner side of the nuclear membrane and provides it with structural support. So in this illustration, what you're seeing is a nucleus of a cell, and the nuclear lamina is shown as this series of green X shapes. So think about the nuclear lamina as being like those metal rods that are on the underside of an umbrella, and they allow the umbrella to stay open. So consequently, abnormalities in the lamin A protein result in a flimsy nuclear membrane that has an abnormal shape. And in these microscope images, you can see a normal cell nucleus on the top, and then the nucleus of a cell from a person affected by Hutchinson-Gilford progeria syndrome on the bottom. And after what we've learned in recent chapters, it should be no surprise that it's difficult for cells to carry out mitosis and cell division when their nucleus is so badly impaired. So the symptoms that arise include poor growth, rapid aging, hair loss, wrinkled skin, kidney failure, loss of eyesight, cardiovascular disease. Interestingly, the brain of children with HGPS appears to develop normally. And so internally, like socially, emotionally, these are children, but their bodies are rapidly aging. The life expectancy of someone with HGPS is 13 to 15 years. So, what mutation causes this disease? The gene for the lamin A protein is located at 1q22. So chromosome 1, Q arm, site 22. The gene there is 57,508 nucleotides long, and at the 1,824th position in this sequence, the HGPS allele has a point mutation. There's a thymine in the disease allele, whereas it's a cytosine in the normal allele. So a C got swapped out for a T. And you may be asking yourself, what's the effect? Is it a missense mutation? Is it a nonsense mutation? And in this case, it is neither. This mutation never even makes it to the ribosome because it creates what's called a cryptic splice site within an exon of the gene. A cryptic splice site is a sequence of mRNA that normally should not be cut. It should remain intact, but a mutation causes it to become targeted by the SNRPs. So let's say this is our mRNA. Here come our SNRPs, and they would normally recognize these cut sites here in the messenger RNA. But maybe changing one of the letters at a different location, so let's say this G over here on the right-hand end, let's say the G turns into a U, and now suddenly the SNRPs think that this is a cut site. So the SNRPs will do what they do. They'll come in, they'll attach, cut, and remove. But now they've removed more than they should. They've taken out a section of the messenger RNA that should have been part of the final protein product. And when this mRNA proceeds to the ribosome, it has a whole section missing. So this is what happens in the case of progeria. The lamin A protein isn't processed correctly because of the point mutation. And therefore, there's important sections missing that prevent the protein from being able to function properly. As with our other two case studies, we'll conclude this one with a little bit of discussion about Sam Burns. Burns was diagnosed with HGPS before the age of 2, and it happened to be the case that both of his parents were pediatricians. So they then founded the Progeria Research Foundation to fund investigations into the cause of progeria and possible treatments. And much of what I just taught you about the cause of progeria was made possible by the Progeria Research Foundation. Burns served as a spokesperson and an advocate for the foundation, but he passed away in 2014 at the age of 17. But while he was alive, the Progeria Research Foundation made significant strides at identifying medications to improve the prognosis of the disease. The organization helped develop a medication called lonafarnib that clears the defective lamin A protein from cells and lowers mortality rate by about 30%. So in the clinical trial of this medication, when they followed up with patients 2 years later, only 3% who were treated with this drug had passed away, while 33% of those who hadn't been treated with it had passed away. So it's no cure, but it does represent a meaningful improvement in the lifespan for children who have this disease. So in our second to last checkpoint here, I'd like you to tell me what would be the consequence of a mutation that removes an mRNA splice site. So in HGPS, we saw how a mutation created a splice site that the SNRPs would recognize. What would be the consequence of a mutation that removes one of those splice sites? So we're basically at the end here. Thank you for coming on that journey with me and learning about these 3 case studies. I tried to give you a few different examples that would give you a sense of the wide variety in ways that mutations can cause problems with proteins and how they can lead to diseases. So hopefully you come away from this lecture feeling like you have a good understanding in a general sense of how genetic diseases work and why nucleic acids and proteins are so important to molecular biology. I'm going to leave you with one final checkpoint to test your overall understanding, and that is, which of these 2 mutations would you expect to be worse for a protein and why? Mutation 1 is ACU changed to AAU. Mutation 2 is ACU changed to CCU. And to answer this question, you'll need both the codon table as well as the amino acid chart, which are both on the screen. And once you finish this checkpoint, you're all done with Chapter 9.